It's a pleasure for me to introduce Dr. Julieta Caballero, who is going to speak to us from Switzerland on in vitro fertilization. Julieta received a Doctor of Veterinary Medicine from the University of Rio Cuarto in Argentina, after which she worked in a private company on embryo transfer and in vitro fertilization on farm animals. She took a summer course in animal reproduction at the Marine Biology Lab in Woods Hole, Massachusetts. She obtained her PhD in animal reproduction from the University of Buenos Aires in Argentina and spent 5 years as a postdoctoral fellow at the University of Laval in Quebec City. Since 2015, she's been working on human in vitro fertilization, first in Toronto and currently in a private company in Basel as a clinical embryologist. She's an active member of the European Society of Human Reproduction and Embryology. She speaks Spanish, English, and French fluently and is currently learning German, since, among other things, her husband happens to be Lucas Seehausen. She's an accomplished equestrian and has won many trophies. Enough of her accolades. Julieta, you're on.
Thank you, Arthur. Thank you so much for this introduction. It is for me a pleasure to be here and to share with you some of my knowledge about fertility and human reproduction. And I hope that you enjoy my talk, and do not hesitate to stop me at any point that you don't understand anything or you would like me to explain a little bit further, or if there's any subject that is of your concern and you would like to discuss it as well. Absolutely, it's a pleasure. I'm not used too much to work with Zoom, so I will just, I don't know how to start sharing my screen, my presentation. Okay, so let's get it started. So my presentation is called Infertility and How Can We Treat It? Oh no, I don't have animation.
I think it was okay.
The screen just come up, Julieta. Next slide.
Okay.
Just a few seconds ago.
Okay, but can you? It's just that I cannot see the animation. But
yeah, your next slide is there.
Yeah. Oh, okay. I will go through like that. So here what we have is a picture of the human karyotype here at your left. The human karyotype is composed by 46 chromosomes, 22 pairs of autosomes. So you can see here, 22, and 1 pair of sex chromosomes. For each pair of chromosomes, one comes from our mom, okay, and the other one comes from our father. Since females can only carry an X as a sex chromosome, it is the sperm who determines the gender of the child, which can be always X from the mom and Y or X from the father. So what is the objective of human reproduction? Well, mainly we have it here, those four. The first is to produce specialized cells, so the sperm and the egg, that are adapted to internal fertilization, that can only contain half of the chromosome numbers in order to reach the correct number of chromosomes after fertilization. And they also have to generate genetic diversity. Those 3 points are accomplished by the egg and the sperm. So the gametes contain half of the chromosomes, and also before getting under this division that we give them half of the genetic material, they experience sort of a recombination between the chromosome from the mother and the father of each of the partners. So those chromosomes that are now contained in the sperm and the egg, they are not the same as the chromosomes that the individual has. Another objective of the human reproduction is to prepare the female body for gestation of the offspring. And this is mainly accomplished by the female reproductive tract together with the embryo and the brain, in some areas of the brain that we will see later. So here we have some facts about the egg and the sperm, some like main difference that is very important to know. So sperm cells are produced in the testes from puberty and during all men's life. But if we compare with the eggs are produced from puberty, also around 14, 15 years old, to menopause. So I would say between 45 and 50, 52 years old. Estrogen hormone and sperm production in the male is a continuous process. So every ejaculate contains about 100 million sperm. So the daily production is massive compared with the egg production. So only one egg is produced per menstrual cycle, and is only produced during the ovulation time. The sperm remains fertile for 2 or 3 days. Sometimes, in some cases, can remain fertile for up to 5 days if they are in the appropriate environment in the female reproductive tract. However, the fertile window of the egg is between 6 and 24 hours. The stem cells that form the sperm are in the testes during all men's life. So men can produce from the stem cells newly produced sperm every approximately 60 to 70 days. But the stem cells for the eggs, to produce eggs, disappear at birth. So women are born with a limited number of eggs, and they will be ovulated or simply go into atresia during the life. But they cannot generate new eggs after birth. Sperm DNA is highly compacted inside the DNA, the nucleus of the sperm. And this is a very specific process that the sperm goes through in order to ensure that the genetic material is safe, because the sperm has to leave the body of the man and reach the egg in women's body. So the cell division in case of the eggs is not completed until the egg is ovulated and fertilized afterwards. So this means that the eggs they stay in certain intermediate stage of the division, and this made them also very prone to some problems in the division later on when the woman is older. And we will see this later on in the presentation. The mitochondria, which is the organelles that produce the energy in our cells, in the case of the sperm, are the only objective is to provide the energy to the sperm to reach the egg. But after fertilization, these organelles disappear. But in contrary, the mitochondria that are in the egg persist in the embryo. And that's why our mitochondria, they only come from our moms and the mom of our mom. And that's why we can track sometimes in genetic testing where our maternal lineage come from, because our mitochondria are the same in all our maternal line. So the sperms are produced in the testes, and the eggs are produced in the ovaries. However, this production is it's a very complex process and is regulated by steroid hormones, testosterone in the case of the testes, and estrogens and progesterones in the ovaries, and many other factors that are a little bit more complex. But more or less, those are the main factors that drive the production of the gametes. However, everything is not finished there. The main regulation of the production of the sperm and the egg comes from the brain. So the brain, certain areas of the brain are in charge to coordinate the production of these steroid hormones and other factors that actually coordinate lately the production of the sperm and the egg. Those areas are in this kind of hidden behind between our nose and our eyes. In this area, especially the hypothalamus and the pituitary, are the areas that regulate the reproduction. What is also very interesting is that those areas in this region of the brain, there are many other systems that are being coordinated. For example, the temperature of our body, the amount of light that we receive, and they coordinate also the circadian cycle, the status of being hungry and satisfied, and when we are thirsty or not. So this is very interesting because many external factors can affect lately or ultimately the regulation of our reproductive cycles. In order to produce a sperm, germ cells in the testes undergo very complex processes that have to take place at 2 to 3 degrees lower than the body temperature. And that's the reason why the testes are outside the body and not the ovaries. So a sperm, because as I explained you before, they become very specialized, have a different membrane composition from other men's cells in the body. And that's why the sperm production takes place inside a very long convoluted tubule. And they are protected there without making any contact with the immune system. And this is to protect them and avoid the immune system to attack them or to recognize them as something not proper from the man's body. Those points are very important because some cases of infertility can come when the testes are exposed to high temperatures or higher than 35 degrees. And also, it is very common to see that when men has been had lesions early in their life, for example, when they play soccer or they play rugby, and sometimes they have some strong impact on the testes. This can produce very small lesions inside and allows the immune system, so actually the blood, to circulate inside the seminiferous tubules, and then the immune system responds against it and produce antibodies. So those antibodies later on can attack their own sperm, and the sperm cannot actually swim properly towards the egg at the moment of the intercourse. What happened to the female counterpart? So the oocytes mature in the ovary. And as I explained you before, they only complete their divisions close to the ovulation and later on after fertilization. They grow in the ovary and specific structures called follicles. And these follicles are getting mature every cycle of the woman, and then only one of them is ovulated during each menstrual cycle. Once the egg is ovulated, the egg falls into the fallopian tubes. And if fertilization occurs, the first 4 days of the embryo development occurs in this fallopian tube. And only after day 5, when the embryo falls into the uterine cavity, the implantation begins. And this happens like this because at the day 5, the embryo has clearly 2 groups of cells. One that is called inner cell mass, that is going to form the baby later on. And another group that is called trophectoderm. And this trophectoderm is the cells, the exterior cells, that are going to form the placenta. And those cells are the ones that are going to interact with the endometrium and start the implantation. So after sexual intercourse, the sperms are swimming up in the uterine cavity, And then once they reach the fallopian tubes, they start to get interactions with the egg, recently ovulated, because remember that the egg remains fertile for only between 6 and 24 hours. So sometimes it happens that the sperms arrive before. And in this situation, there are some hormonal changes that keep the sperm fertile during this time, waiting for the ovulation. But if the ovulation has occurred, the sperm start to undergo some changes in the membrane that allows the sperm to make contact with the egg. And once they reach the egg, they will be able to go through the zona pellucida, which is a shell that covers the egg. And once the egg is penetrated, the egg triggers a signal that blocks the membrane, and then not any other sperm can enter. So only one of them will stay inside. After the sperm is in the cytoplasm, the DNA starts to be decondensed, and this also stimulates the egg to complete the division and to also start to decondense the DNA. And then occurs the syngamy, which is the encounter of the two sets of chromosomes from both gametes, and the embryo starts. After this, there's a couple of days of just cell division until some of those cells start to be committed to form different groups of cells, and the more complex embryo development starts. So I will explain here a little bit of the physiology of the reproductions in humans. And now, so let's go to see what happened when a couple cannot conceive. So, most of the times they realize that it's a problem when they have been trying to conceive and, they cannot. But sometimes they also don't know how much time they should wait until, they can conceive or not. So, World Health Organization has defined infertility as a disease of the reproductive system, defined by the failure to achieve clinical pregnancy after 12 months or more of regular and protected sexual intercourse. But this has to be, we have to also consider some other points, which is that the couple has to be, of course, in the reproductive age, which means between approximately 15 and 49 years old, but usually 45 is the limit. That they also, sometimes the problem is not to conceive, but to keep the pregnancy going or give birth to have a live birth. We also have to consider that they have been not using any contraceptive method, that the woman is not in the lactation process. And of course, they are keeping a desire to have a child. So which are the causes of infertility? As I explained you before in the general introduction, so we have two parts, the male and the female, that they have to work properly in order to reach, to succeed. So causes of infertility can be separated in two main groups. I would say three main groups. So of course, male factor causes and female factor causes, and some of generated by lifestyle. So in the case of the male, any factors that affect the production of the hormones or any genetic factors that can affect the production of the sperm or that affect the motility of the sperm, for example, some medications or drugs, or also some obstructions of those tubules that I showed you before, can cause infertility. Age as well can cause infertility, but it's not as important as in a woman because males can produce millions of sperm, and certainly somehow few of them are in good shape, are sufficient to produce a baby. However, those problems are a little bit more complicated in the female side because the female can only ovulate one egg per month during certain years. So one of the main factors of infertility in women is age. because of course, as women age after they are 35 or 38 years old, fertility declines just because the eggs are not as fertile as before, or the hormonal regulation is not what it used to be. There can be also some ovarian factors, some diseases that can affect the ovulation or the production of the estrogen hormones that are very important to also maintain the gestation as a progesterone. But of course, as in the male, the female can also have anatomical problems that can cause infertility, like when they have the fallopian tubes blocked, or there's some cervical malformations, or the uterus is simply not able to have a proper lining to receive the embryo. But there are factors that also affect both partners that are related to the lifestyle, like sedentarism, overweight, smoking, or alcohol, or just some drugs, or also especially in the last century, pollution and stress is one of the main causes also of infertility. Data from the European Society of Human Reproduction and Endocrinology in 2016 said that 1 in 6 couples worldwide experience some form of infertility problems, and at least once during their reproductive lifetime. 20 to 30% of infertility cases are explained by physiological causes in men, and between 20 and 35 by physiological causes in women. And between 25 and 40% of cases are because of problems in both partners at the same time. There is also very important, especially in the last, I would say, 10 years or 20 years, there's 10 or 20% of causes that cannot be found. And there's actually many groups doing a lot of research about it. Infertility, as I explained before, can be associated with simple lifestyle factors. And one of the most important causes that we are treating every day is advanced maternal age. So actually, advanced maternal age causes aneuploidies in the embryo. Aneuploidies mean an abnormal number of chromosomes in the embryo. And this is caused in the embryo because of the abnormal chromosomes already in the egg. And as what happened is that with the chromosome number is not appropriate, the embryo will go, so the woman will get pregnant, but the pregnancy will not go further than 12 weeks. And this is called early abortion, we call it miscarriage. And as you can see here in this graphic, as the women age, fertility declines at the same moment that miscarriages increases. So this is actually one of the most common causes of infertility we have and we treat every day. But it's also very interesting. I want to share with you this information. There was this paper that was published last year. They did an analysis from men samples from more than 40,000 men in 50 different countries in the North Hemisphere between the 70s and 2011. And this study revealed that the sperm concentration has declined more than 50% during this time. And so it means that many other factors like stress or pollution that can affect it also, that can affect the production of the sperm in the testes. Actually, there are many works that analyze the incidence of infertility of certain plastics or chemicals that they are used to the production is using the chain of the production of plastics. So what can we do to help those couples to conceive in the IVF clinic? So as many of you may know, already in 1978, the first baby was born by in vitro fertilization, or what we call most commonly as a test tube baby in England. And after her, millions of other babies have been born from the same technique. At the beginning, there were some concerns because the technique could cause some problems, not in the early years, but maybe later on in the life of these people. But actually, what we have seen is they're absolutely normal. And if any issues or diseases are presented later on, they're actually at the same incidence that in people that have been conceived naturally. Actually, not so many years ago, Louis Brown was able to conceive his first baby, but naturally. So this demonstrates that actually she is fertile and nothing is carry, and not any problem is carried over the next generations. So here I'm going to explain you a little bit what we do daily in the clinic when we receive a couple that wants to undergo a fertility treatment, what we call an IVF treatment. So the first thing that we do is to, of course, review the medical history and to assess the hormonal levels, ovarian function, and endometrial status in the woman. And at the same time, we ask her partner to provide us with a sample, and we evaluate sperm parameters, basically the total amount of sperm and how many of them are normal morphologically and they can move forward. Then when we have an idea of what's going on, we have probably a diagnosis or probably not. So we discuss with the couple the possibilities to go under the treatment. And the treatment starts when the woman is being stimulated with hormones to produce more than one egg. We do this because, of course, if we only recover one egg as naturally the woman ovulates, the chances are very low that we can give them back an embryo. So in order to do that, we put the woman in a treatment during, depending on the time of the treatment, the type of treatment, during 1 or 2 weeks. And then at the end of this moment, we induce the maturation of the egg with hormones similar to LH, as luteinizer hormone. And before the eggs are ovulated, we collect them by aspiration of follicular fluid. Then as embryologists in the laboratory, we search for those eggs, we classify them, we analyze them, and those that are mature, we are gonna fertilize them in vitro. On the other hand, there is another group of technicians in the lab that are searching for the best sperm possible. The selection is being done based on the best motile and morphologically normal sperms. But sometimes may happen that the man doesn't have any sperm in the ejaculate, with what we call azoospermia. And in those cases, we have to perform a biopsy of the testes, and we recover some of the sperm that actually, if they are immature, it's not a big problem because we only need the DNA. And what we do is we inject the sperm inside the egg, and then so we bypass all of those interaction processes that naturally sperm do to fertilize the egg. So in vitro fertilization can be performed in 2 different ways. So what we call IVF or what we call ICSI. IVF is simply just we put in contact the sperm with the egg if the sperms are motile. And we wait for a couple of hours, then we remove them and we check next day if those eggs have been fertilized. In the case of ICSI, we usually use this technique when the sperm cannot reach naturally the egg for some issue, and so we inject them directly inside the egg. So as you can see in this picture, so with one needle we hold the egg, and with another very tiny needle, we pick up one sperm and we inject it directly inside the cytoplasm of the egg. Once we have those eggs that have been fertilized and we culture them in vitro, it means in dishes in very controlled conditions in the lab during 5 or 6 days. And then we choose one of those to transfer in the uterus of the woman. However, sometimes it might be that because of women's age or because of other genetic issues, we need to test genetically those embryos before transferring. In those cases, we freeze all the eggs and we biopsy them and we send the samples to a genetic lab. And once we have the results, then we can thaw those embryos and transfer that embryo that is normal. So when I talk about pre-implantation genetic testing, what we do is here we have a picture of an embryo, and we go with a small needle and we pick up some cells from the embryo and we send it to the genetic lab. Those cells are not random cells. We take cells that belong to a group of cells called trophectoderm. And if you remember, I explained you before, those are the cells that are gonna become the placenta. So those cells have no impact on the development of the embryo later on. So we never touch the group of cells that are gonna later on give place to the embryo. So once we have the genetic information from the lab, we thaw one of those embryos and we transfer in the uterus of the woman. And then we have to wait approximately 7 to 10 days to do the pregnancy test. We don't do regular pregnancy tests like those we find in the pharmacy. We take a blood sample from the patient and we assess the hormones directly, so to be more specific. If the woman is pregnant, then she will go through a set of scans that we normally do with our women that are pregnant naturally. And once the woman passed the week 12 or 13, so that we don't have any more possibilities of miscarriage, the woman is transferred to their own doctors or obstetrician. So what is the successful rate of those treatments? Well, many people think that actually going to the IVF clinic means I go there and I come back home with a baby, but actually, and unfortunately, this is not true. So it's still a technique that is very inefficient. So the mean pregnancy rate per embryo transfer is around 35%. And this is higher when the woman, of course, is young. And when the woman passes the 35 or the 38, this is getting a little bit more difficult. And the pregnancy rate per treatment is about 30%. And the delivery rate per treatment is around 20%. So, and that's why it's very important that we take many eggs per treatment so we can have more chances. But also it's a good balance because sometimes too many eggs is poor quality. So everything has to be assessed from the very beginning regarding how many follicles the woman is having at the beginning of the treatment. So when we perform pre-implantation genetic testing for aneuploidies, so for abnormal number of chromosomes, these chances can increase up to 60%, which means most of the times we have embryos to transfer, but they don't implant simply because they don't have the proper number of chromosomes. So I think I have been explaining a little bit what we usually do in our jobs in the clinic. If you have any questions or something that was not clear or something that you would like to know a little bit more about, you're just free to ask.
Julieta, maybe you can stop sharing your screen, please.
Yes. Yeah.
Okay.
I have one thing here, if I may. Let me just. there. Just hang on 1 second. Can I just share something?
Yes.
Here. Where are we here? Can you see this one here? This is kind of what I thought how it works. How the human conceive.
Yeah.
It's not quite like this, I guess. Okay. Thank you.
Yeah.
Okay, we have time for maybe 2 questions, and that'll be it because we're running a bit over time here. Alan, you got your hand up. Go ahead.
Yeah, I assume there's a cost to this in fertilization in vitro. What's the average cost? Does it vary from country to country, or is it standard across the board?
Yeah, it's really, it's very different from country to country. Actually, what is also different is what the insurances cover. So, for example, in Canada, they used to cover 1 or 2 treatments when the woman was 38 or more, but not for younger couples. Here you have to pay it yourself, and it can cost, for example, here in Switzerland, between 20 to 40,000 francs. It depends on all the add-ons. If you want to do genetic testing, or if somehow it happens, for example, in 1 cycle we don't have embryos, so we have to do a second treatment. And sometimes we have many embryos, so the woman has to go through many embryo transfers. So every embryo transfer costs more as well. But I will say that around 30,000 francs or dollars, approximately.
Okay, any more?
Yeah, I have 1 question here.
Okay, Danny, sorry, Danny, this will be the last question. Sorry, go ahead, Danny.
You were saying about the stimulation of the ovary to produce the eggs. What kind of drugs do they use, and what are the impacts on the individual after it's all over?
Yeah, so mostly we use hormones, those hormones that come from the brain, as I explained. So those hormones that gives the orders actually to the ovary to produce estrogens, progesterone, and start to select the eggs that are gonna mature. So those hormones are can be synthetic or can be purified from. Actually, some of them are purified from a menopausal woman, because when a woman enters in the menopause, they start to produce a lot of those hormones from the brain, trying to stimulate the ovaries, and the ovary doesn't respond. So in women's urine or blood, we can find these hormones, and we use it for the treatment. Those hormones, sometimes what happens is that when we start the stimulation and there are many follicles, so many eggs that are recruited, so for example, more than 20, there's a high production of estrogens in the ovary. And those estrogens can produce a syndrome that is called overstimulation syndrome. And in those cases, we have to most of the time stop the treatment, and the woman has to be hospitalized or sometimes being treated by the doctor, because they can have very bad impacts on the health of the woman. But nowadays, the hormones that we are using has been changed molecularly. So the stimulation that they produce is controlled. And those syndromes we see, but I will see in 1% of the cycles that we have.
Okay.
Okay, thank you, Julieta. Alan, you're scheduled to formally thank Julieta, please.
Okay, that's nice to know. I forgot to read my notes. Well, Julieta, of course, as usual, when you and someone like Arthur speak to us, it's very, very complicated for someone like me to understand it. But we do understand that, you know, that there's test tube babies. We read about it because we're all old enough to remember these first test tube babies from years ago. And it's been great having you to come out and explain it to us uneducated people maybe some of the women understand us better than the men. But thanks again for taking your time out to speak to us today. Thank you, Julieta.
Thank you for having me here. It's a pleasure.
Okay, now, Julieta, you're welcome to stay on if you want to, but if you've got to go, we quite understand. So it's entirely up to you.
Okay, yes, I will leave you so you can discuss and go on with your business.
Okay, all right. Thank you very much.
Thank you. Have a great evening.
Thank you.
Julieta.
Bye-bye.
Thank you as well.